So today's video is going to focus on organic compounds and the monomers, the polymers that make them up, and their different jobs or roles in living organisms. We've already talked some about DNA, so we won't spend a lot of time on that one. We'll focus on the other three major categories more in this particular video. Remember, these are your notes. Write down anything that you feel will help you, draw pictures of things if that will help you, right? Remember, personalize them, make them work for you. Okay, so as we talk about organic compounds, we're talking about compounds that are going to contain carbon-hydrogen bonds. So that's what's going to make it an organic compound or a biomolecule. Is it's got to have a carbon-hydrogen bond. It can't have just carbon. Carbon is going to be important to us because while it's not the most abundant element that I'm going to find in a living organism, I'm going to find more hydrogen and more oxygen usually. It's third in line there. And as you'll see in our next picture, it is able to do a lot of different kinds of bonding, so form lots of different combinations. So we're going to be talking about organic compounds, which means they all have a carbon-hydrogen bond. So for example, CO2, carbon dioxide, would not be an organic compound because it does not have a carbon-hydrogen bond. So you'll see in this image here, carbon is able to form multiple bonds. This example here, I've got lots of carbon-hydrogen bonds, so definitely an organic compound here. It's a better building block than hydrogen because hydrogen can only form one bond. It's a better building block than oxygen, mainly, again, because carbon, like I said, is able to form 4 bonds. So we're going to be looking at the different varieties of these organic compounds that can come from these carbon-hydrogen bonds. So let's shift gears over to these specific macromolecules. Macro means large. So these are going to be these large polymer building block of life molecules, not elements, not just carbon, not just oxygen. We're going to be talking things like proteins, carbohydrates, lipids, nucleic acids. So there are 4 major classes of macromolecules, or you'll see them referred to a lot as organic compounds. So a lot of the times in star materials and such, these two things will be interchangeable. So there are 4 major classes of them. Carbohydrates, which essentially is our sugars. Lipids, which is our different types of fats and waxes. Proteins, and then nucleic acids. Nucleic acids are our DNA and RNA. We've spent a lot of time on DNA already. We'll touch on RNA a little bit, but then we'll revisit both of those when we get to DNA replication and protein synthesis. So let's look at each one of these specifically now. This unit is going to have a lot of memorization, so please make sure that you're getting this information written down so you have it as a resource when it's time to study. Okay, before we jump too much into specifics, let's revisit monomers and polymers again. So we talked about these in our DNA video. Remember, polymers are made of many monomers linked together. The monomer is going to be the building block, and the various organic compounds are going to have different polymers, different polymer names, and different monomers, and that'll be something you'll be expected to know. So we can both build these polymers up as well as break them back down. So let's look specifically at building up a polymer. So synthesis, right? I'm making or I'm building something. So when I build up a polymer, one of our most common reactions that does that is called a dehydration synthesis reaction. So this dehydration synthesis, when I dehydrate something, right, I'm going to lose water, and I'm losing water to make or assemble a polymer here. You can see that happening in the picture over here to the right. Enzymes, we'll talk more specifically about enzymes in our next video, but enzymes essentially control chemical reactions. So enzymes essentially control chemical reactions. So an enzyme is going to help remove this water. You can see how water is being lost here. And my end result here now is I get this fully synthesized polymer down here below, each circle representing a monomer. And I'm using this removal of water controlled by that enzyme to bring it together into a larger polymer. So that would be a dehydration synthesis. This is an example of an anabolic reaction, which is going to be a building type of chemical reaction. Our next example here, hydrolysis, is a digestion or a example of a catabolic reaction, which would be a breaking down type of reaction. So hydro is water. Lysis is to break apart or to break down, to break. So in this case, I'm doing the opposite here. I have this large polymer here, and I'm adding water to that polymer, also using an enzyme because the enzymes control chemical reactions. And now I'm breaking this into separate monomers and breaking the polymer up. So catabolic hydrolysis reaction is going to add water to break apart the bonds, where that dehydration synthesis removes water to build the polymer. A lot of your names in biology, they're going to give you the answer. So when we do vocabulary or we put a lot of emphasis on vocabulary like this, make sure you get it down, file it away, because it will make a big difference. It will help you a lot. For instance, this lysis term is going to show up multiple times throughout the year. And if you can remember that lysis means to break, you're going to be able to figure out what stuff is. Okay, let's look specifically at carbohydrates now. Carbohydrates are going to contain the elements carbon, hydrogen, and from now on, these will only be abbreviated, and oxygen. These elements are going to be usually found in a 1-to-1 ratio. You'll see here, if I simplify, this example that I have here is glucose. And if I simplify that down, 1-to-1, carbon, hydrogen, oxygen. But there's nothing else. So if you see a nitrogen, if you see a phosphate, it can't be a carbohydrate. It can only contain carbon, hydrogen, oxygen. Their main functions of carbohydrates is quick, short-term energy, right? Again, remember that these are sugars. And that's a common thing for sugar, right? Is that we think about it as that quick energy boost. And once we link these into much longer polymers, into these longer sugars, they can be used to help with structure and storage of things. The monomer for a carbohydrate is a sugar, which is called a monosaccharide. I spelled that wrong. I should say monosaccharide. Saccharide, when you see that, you need to be thinking sugar. Okay, the monosaccharide, the mono part, mono means one. We're usually referring to the ring structure of these carbohydrates. Carbohydrates will usually have a ring shape. If you remember back to DNA, remember that 5-carbon sugar, that's considered a ring shape. Okay, you could have a 6-carbon sugar. Right, so that ring shape, if you see an image and you see it shaped like that with a ring and it only has corners, right? Remember, there's each corner represents a carbon, right? Each corner is a carbon. So as long as there's no N's in there, no P's, no S's, right? You know that they're all carbons. Therefore, you can most likely assume that this is going to be a sugar molecule. Okay, let's look at some examples of these. You can see monosaccharides. Here's an example over on the side here with our glucose. Here's my ring structure that it has, right? This overall ring structure here. It is a monosaccharide. Some other examples of monosaccharides other than glucose would be fructose, which is the sugar we find in fruit, okay, and galactose. Disaccharides, di means two. So in a disaccharide, we're going to have two of those monosaccharides linked together, but no more than two, right? So I'm not going to have a big, long, giant chain. So some examples here would be sucrose. Sucrose is table sugar. It's what you cook and bake with, and granulated sugar. Maltose would be an example, as well as lactose, the sugar we find in milk. Polysaccharides, remember poly means many, saccharide means sugar, so many sugars. These are going to be super long chains. Like there's no way I can draw all of this. Or hundreds of sugar molecules linked together, possibly thousands of sugar molecules linked together. So some examples here would be cellulose, chitin, glycogen, and starch. So these are the ones, these polysaccharides, these are the ones that can be more helpful in structure as well as storage. So cellulose is what we see in the plant cell wall. So that's obviously a structural carbohydrate. Glycogen and starch are used for storage of extra sugar. Glycogen is storage in animal cells. Starch is storage in plant cells, hence the potatoes that are there. So we're storing the extra sugar. If we run out of room to store it in the form of glycogen in an animal, we then store it as fat. Chitin is another structural one. It's in the cell wall of fungus. So it's another structural polysaccharide in the cell wall of fungus. Hopefully you've noticed a common suffix here that may help you. If you come across an unknown molecule, like maybe you don't have all these memorized, but you see this ose, right? You come across an unknown molecule and you see that ose suffix, you need to be thinking that it's a carbohydrate or a sugar. Okay, our exceptions, right? Glycogen doesn't have that, starch doesn't have that, chitin doesn't have that, but you notice everything else does. Cellulose, sucrose, maltose, lactose, glucose, fructose, galactose, right? So keep that suffix in mind. Okay, let's switch gears over to lipids. Okay, so lipids are going to have some things that they have in common with carbohydrates in the sense that they have the same elements. So I'm composed of carbon, hydrogen, oxygen. However, remember for carbohydrates, those were usually in that 1-to-1 ratio, and they were usually formed into a carbon ring. Lipids, that is not the case, not the 1-to-1 ratio, and not necessarily in a ring. So our functions of lipids, I'm going to have long-term energy storage. Remember, carbohydrates did short-term. So I'm about to run a race. I don't want to eat fat a minute before the race. I want to eat some glucose or sugar a minute before the race. But I'm going to run a marathon a day or two before the race. I might want to make sure I have a little bit of fat for some long-term energy storage. Fat is also lipids are also going to help with insulation and protection, right? I know most of you have heard of whales and seals, how they use blubber to help them keep warm. The protection, for example, in a human, our kidneys don't have a lot of bony protection, so they're embedded and surrounded by fat to help protect our kidneys. So lipids can do that for us as well. Lipids also are important with regulation and cell signaling, as in cell communication. One cell in your body telling another cell what to do. We have two basic types of hormones, which are essentially cell signals in our body. We have protein-based hormones and lipid-based hormones. So really important for regulation, in particular for regulation of male, female, secondary sex characteristics. Those are all lipid-based hormones. And then they also can be important for structure, in particular in our cell membrane. And we're going to look at examples of each one of these a little bit more specifically. But make sure you have these 4 functions down: long-term energy storage, insulation and protection, regulation and cell signaling or hormones, and then structure. So let's look at some examples of lipids. You'll notice we have not said this is the specific monomer of a lipid because it doesn't exist. Where carbohydrates have that specific monosaccharide, and I link a bunch of monosaccharides together, I'll get a different kind of polysaccharide. I link a bunch of glucose together in an animal cell, I'll get a glycogen. That's not how lipids work. While they are a polymer in the sense that they're a macromolecule made of lots of smaller molecules, there's a wide variety of lipids and they don't have a specific monomer. So let's look at our first one here, a phospholipid. Phospholipids are crucial in cell membrane structure. And this is a phospholipid there. When we talk about the cell membrane, we'll talk more specifically about the phospholipid. Hopefully you've gotten from the name that it's a phosphate group attached to a lipid. And that is really going to dictate the structure of our cell membrane, which will dictate how it's able to work. Okay, some other examples of lipids are this steroid hormone here. Remember, this was for our cell signaling and regulation. We also have a triglyceride, tri meaning 3, right? 1, 2, 3. And then it's got these fatty acids attached to it. Sometimes you'll see this mentioned as a long E structure because it'll look kind of like that when it's sketched out. So in addition to these, obviously fats would count, waxes would count as well. For instance, again, if we're going to talk about a multicellular organism, let's talk about us, right? We produce a wax, we produce earwax. Okay, that's a lipid-based molecule. So phospholipids, steroid hormones, triglycerides, fats, waxes, wide variety of examples here. And our last thing to talk about with lipids here are saturated versus unsaturated lipids. So a saturated lipid is going to be solid at room temperature. You can see with the butter and stuff. So solid at room temperature. An unsaturated lipid is going to be liquid at room temperature. Vegetable oil, olive oil. Okay, so if we look at this zoomed in picture here of the actual molecules, you'll see how this one's nice and flat, but then this one is bent here. You'll notice there is 2 lines between those carbons there. That represents a double bond. And so a saturated fat has only single bonds, where a unsaturated fat has at least 1 double bond. And what this does is it allows these molecules to kind of, they're flexible, they're bend, they shift around. And so when there's only single bonds, it allows them to pack nice and tightly together, which is what lets them be solid at room temperature. When there's a double bond in there and I get this bend, they can't pack. Like, see how these could kind of pack nicely together and be solid at room temperature? When they have that bend in them, then they can't pack as tightly together because that bend pushes them apart some, so they stay liquid at room temperature. Okay, let's shift gears to proteins. I know we've thrown a lot of information. You're going to have to keep reviewing and reviewing these because it's, a little bit of detail and it's just memorization. We'll practice with it some in class, but you're going to have to do some on your own. So remember, your focus is going to be at minimum the monomer, the name of the polymer, if parts make up the monomer, and then the function of the molecule, and then be able to compare those to one another. So let's look at proteins. It's our third one. Our fourth one we've already talked about a lot because we've talked about DNA a lot. Okay, so our third one here is a protein. It's got a monomer here. That's the amino acid. Okay, so the monomer of the protein is the amino acid. That's this image right here. This is an amino. That's one amino acid. Proteins are crucial. They essentially dictate everything for organisms, multicellular organisms, single cell organisms. They dictate it all. Amino acids have this NCC pattern. So where before in carbohydrates and in lipids, we only had carbon, hydrogen, oxygen. Proteins are definitely going to have nitrogen in them as well. So that's a giveaway. If it's a nitrogen, it can't be carbon, it can't be lipid, it could be a protein, or it could be a nucleic acid. Sometimes they'll have sulfur, sometimes they'll have phosphate, they'll have all different kinds of other things, okay? But they'll definitely have a nitrogen. Each amino acid is set up in this NCC pattern that you can see here in this picture at the bottom, right? In the sense that I've got a nitrogen, a carbon. So they'll always be in that pattern. Coming off that middle atom there, off that carbon, you'll see this R group, or what's called the variable group. That's what makes each amino acid different. If you look at the picture on the right with the two amino acids that are going to join together, sorry, this water molecule should be over here. There's the water over here. You can see how both amino acids have that NCC pattern. Okay, the R group is what would make them different. Okay, that's the only thing that would make them different. And when I join those two amino acids together, remember I'm losing water and I'm making something. So this is an example of a dehydration synthesis reaction. I make the polymer, which is the polypeptide. Poly means many. I make those peptide bonds. That's what holds those amino acids together, is a peptide bond. So a protein is usually a synonym for a protein is a polypeptide, with lots and lots of amino acids linked together. And again, we'll look closer at that polypeptide term when we build proteins, when we do actual protein synthesis. Right now, the minimum I need you to have is that the monomer is an amino acid. That amino acid has that NCC pattern, with that R, or that variable group, coming off that middle carbon, making those amino acids different. I can use a dehydration synthesis to link them together into a polypeptide, which is that bigger polymer. Okay, we're not going to spend a lot of time here on the structure of a protein. That's a thing we'll jump to in AP biology. But I just want you to really get the idea of how many amino acid monomers are linked together to form this big polymer. Each one of these little circles here in this primary structure is an amino acid. And you'll notice it's fading here. You can't even see the circles anymore. Still made of tons of circles. So proteins are made of thousands of amino acids linked together. Okay, so lots and lots of these monomers are required to build this bigger polymer. Okay, so let's look at some functions of protein. You notice the pictures of these proteins look kind of like a big mess because they are so long. Because they're so long, they can't stay just in a big long chain. They have to be twisted and folded and bent. And their structure of the protein, this will be important, enzymes, their structure determines their function. So exactly how they are folded and bent is not just crazy. It's on purpose, and it's a certain way, and it's a certain configuration. And if they're bent, like let's say these kinks are wrong, let's say they're a straight line, well, this protein now doesn't work. It would be broken. Okay, so their structure is very, very important. So proteins have a wide variety of functions. They can, again, help with storage. They can help with structure. In particular, if you think like hair and nails, the protein in those that gives those some structure is keratin protein. Movement, muscle cells are made heavily of proteins that allow for movement. Some of our immune things like antibodies are made of proteins. Enzymes we've mentioned before that they control chemical reactions. That's going to be our next video, is exactly how those work. And again, like we mentioned with lipids, we've got hormones that control cell signaling and cell regulation. And like I said, we had two basic kinds: protein-based hormones that control information and steroid-based hormones that control information. So wide variety of functions. Go ahead and try to get those down. Okay, nucleic acids are going to be very brief. We're going to talk about them again when we talk about protein synthesis. And we've already talked heavily about DNA. So the expectation is that you remember a lot of this. Okay, so our two nucleic acids, we've got DNA, which is that we've already done, and we have RNA, which we'll do more in depth when we do protein synthesis. So DNA, or deoxyribonucleic acid, and RNA. So the main job of DNA is to store the genetic information. So DNA, deoxyribonucleic acid. And then we have RNA, or ribonucleic acid. That's based off the sugars that are in them. It's going to transfer information for us, because remember, DNA is always going to be found in the nucleus. It can't leave. It's trapped in the nucleus. And so in cells that need it to leave the nucleus, they need RNA to help transfer that information for them. Okay, so as we've mentioned before, the structure of nucleic acid, the monomer is a nucleotide. Remember, a nucleotide is going to have that 5-carbon sugar, deoxyribose or ribose, the phosphate, and the nitrogen base. Remember, DNA is going to be double-stranded. That's our double helix that we see down here. RNA is single-stranded. Again, we'll do more detail with this, but get some down now, right? RNA is single-stranded. DNA has adenine, cytosine, guanine, and thymine. These are the nitrogen bases. RNA has adenine, cytosine, guanine, uracil. Remember our purines versus pyrimidines. Pyrimidines are cut from purines. So pyrimidine, right? They're cut from purines. DNA is going to store our genetic information. RNA is going to help transfer that information. Okay, and so if you need to revisit your DNA notes to get some more reminders on structure and function of DNA. Okay, so as mentioned in our previous video, please have made notes of any questions within your notes. Circle, star, write it off to the side so that you don't forget, so that we can clear anything up before we start jumping into labs and various activities with this material.